In this part, we're gonna see meiosis 1, the first division that helps create genetically unique sex cells. In prophase 1, chromosomes start to condense and become visible under a microscope, but there's what makes this phase special. Homologous chromosomes pair up, forming tetrads. Then comes the magic, crossing over. The chromosomes swap little pieces of DNA, which increases genetic diversity. The nuclear envelope also begins to disappear, and the spindle starts to form. In next is metaphase 1. The tetrads line up right in the middle of the cell, spindle fibers attached to each homologous chromosome, getting ready to pull them apart.
In anaphase 1, the homologous chromosomes are pulled to the opposite sides of the cells. Remember, the sister chromatids stay together. This is different from mitosis. Finally, in telophase 1, the chromosomes reach the cell's poles and two new haploid cells start to form. Each has half of the numbers of chromosomes. Sometimes the nuclear envelope comes back briefly and then, boom, the cell splits throughout cytokinesis.
Meiosis II is the second division that separates sister chromatids that may not be identical due to crossing over in prophase 1. In prophase 1, chromosomes condense, nuclear membrane dissolves, centrosomes move to the opposite poles, perpendicular to before. Then, in the metaphase 2, spindle fibers from crossing centrosomes attach to chromosomes and they align along the cell equator. Then, in anaphase 2, the spindle fibers contract and separate the sister chromatids. They are now called chromosomes and they move to opposite poles. For the last, in telophase 2, the chromosomes decondense, the nuclear membrane reforms, and the cells divide, the cytokinesis, to form 4 haploid daughter cells. Wow! And that is the final outcome of meiosis II.
The crossing over occurs in the nucleus envelope breaks down the chromatic contents into chromosomes. Homologous chromosomes containing the two chromatoids come together to form tetrads, joining at their centromes. This is when crossing over occurs, which creates genetic variation. Random assortment occurs at metaphase 1. Homologous pairs will line up in the middle of the cell completely randomly. The orientation of one pair will not affect the orientation of the other.